Thanks for coming, everybody. I'm Mitch Frankel. I came out of the university here, graduated in 2013 from Dick Norman and Greg Clark's laboratories. I've worked at a few of the companies here in the Valley, Ripple, then BlackRock, and then I've been at Epitel since 2017, really spearheading a lot of our data science and algorithms. And that's a lot of what I'm going to talk about here today. Let's see. So before I get going, I wanted to sort of take care of some definitions. We're going to talk a lot about EEG today. And a lot of people, when we say EEG, we think about these long dangling wires and these big bulky systems. They'll have a screen and some squiggly lines running across that has some brain data being shown. There's also what I'm going to talk about is ambulatory EEG. And this is where we can have these recordings in sort of an at-home environment, and it still is these big, long, dangling wires, electrodes coming from the scalp, going to a box that someone's got to sort of carry around and wear with them. But when we talk about what actually is EEG, and when I say EEG, I mean electroencephalography, or an electroencephalogram. And the Mayo Clinic has a really nice, simple definition. And it's that an EEG measures the electrical signals coming from the brain. And that sounds really nice and simple. And if we're talking about more of like implanted, like intracortical EEG, that's really true. But these more scalp systems, the scalp recordings, it's not so much. Really, we're recording from around the brain. And there's a difference. There's a lot of other activity that we're picking up. It's really just general electrical activity that the EEG is recording. This can be noise from the lights. This can be electromyography from muscles. Think about like eyes, the eye blinks. These can be picked up. They're much orders of magnitude larger than brain activity. Brain activity is really small, down on the order of microvolts. And this can really confound a lot of analysis that we have to do from a lot of these artifacts. So scalp EEG presents a lot of difficulties for us from analysis. And I'm going to talk some about that here as we go. This, however, is my favorite EEG system of all time. So I just want to talk about what is a seizure. Most people, when you say the word seizure, this is what they think of, is somebody on the ground having convulsions, they've lost consciousness, they're unable to control anything, and something that lasts for a long time, and they're really kind of at danger of hurting themselves. But that's not really the case in all seizures. This is sort of one very specific type. This is what we call tonic-clonic seizures. You used to hear it called grand mal before. It's really less than half of all seizures that occur out there. There's many other seizure types that occur. Some can be very focal to very localized regions of the brain, and you might see activity just on certain areas. Somebody might have an eye twitch, somebody might smell something, they might have an aura, they might have a taste. There's others that happen most commonly in children called absence, where they stare off, they just kind of blank out for a second. Some happen where they're really just localized to the brain, and you don't actually see any outward symptoms. There's no clinical manifestation. They can be very short. There may be nothing obvious at all to the person or to observers. And really, there's a wide gamut of what a seizure can be. It's not just what you're used to seeing and expect. Really, there's a merging. It takes both the clinical symptoms and being able to see it on the EEG to be able to confirm what kind of seizure it is and that it actually is a seizure. Just because a kid stares off into space doesn't mean they're having a seizure. Just because someone's shaking doesn't mean they're having a seizure. It really takes both aspects. So these are different clinical definitions as well as what it looks like in the EEG. And it's not necessarily one-to-one. On the bottom left, it's about a 3 hertz, we call a spike and slow wave. And yes, it's very common for this typical absence activity, but it doesn't mean that someone's, having an absent seizure. We can see it in other activity. As well, we talk this idea of a focal seizure. It's happening just very localized to specific regions of the brain. We might see it just on certain electrodes. But we can also have it where we call it subclinical, where we don't actually see any outward manifestations. There's no clinical aspect to it. But if I were to look at on just the EEG, I don't know. I need the clinical manifestations as well. So it takes both aspects to really put these definitions to it. Okay, that was sort of the defining part. I'm going to talk a little bit about the company, where we started and how we got to where we are. Because what we're doing now is not what they started from the beginning. And the people that created Epitel are actually no longer with us today. And it's kind of an interesting story of how we evolved through all the years. Epitel started in 2007. It came out of Ed Dudek, a professor here, out of his laboratory, as a partnership with the University of Utah Research Foundation, as well as the anti-convulsant screening program. And this was a large program here at the university where these medications that needed to be tested for epilepsy drugs, they were doing small animal studies, but most of the time they would evolve into going to human trials. And the way that a drug works or doesn't, whether someone's having more or less seizures, would be paper diaries. People would just write down, when they're having seizures or not. And as you can imagine, people aren't very accurate with this. They're not very good at writing these down. People don't always remember when they're having them. And sometimes they just even forget to write them down. It's just not the best accurate outcome measure. So they really wanted a way to do better drug development for all of these anti-seizure medications. And the idea was really, could they come up with some kind of really long duration rodent studies where they could record EEG over very long durations. And really the only way you could do that at the time is have implanted electrodes coming up through these wires to a commutator and a tether. And it's just not a way that you could do very longevity studies. So the idea was come up with some wireless implantable EEG sensor. And that led to the development and eventually the product launch of this product called Epic. And that was Epitel's original product. And it's this wireless transmitter that could be implanted in small animals, the small rodents. And the way that this worked is it generated an electrical field and it capacitively coupled EEG onto that electrical field. And from there, you could have a tray underneath the animal cage and it picked up this electrical field. And then you could strip the EEG off, export it out over a BNC cable, and then put that into any kind of data acquisition system. And in this way, you can have a lot of animal studies. You could, be able to test medications over the lifetime of an animal. The animal's able to be freely behaving and really be able to do a lot higher throughput of these anti-seizure medications. And it was a really great product and, worked really well for this one application. The unfortunate side is it didn't have a lot of use outside of this one laboratory and a few others out there. There really just wasn't a long-term market for us to continue manufacturing this. And we eventually spun this product off. And the people at the laboratory for Dudek, this is what they really wanted to be part of. And we spun them and this product off from Epitel. But this really got everybody thinking. We could do this for small animals. What about wireless EEG for humans? Could we take the same idea and could we create some kind of wireless wearable EEG that allows freely behaving humans? We look at this idea of ambulatory EEG. People have to, have these big, long dangling wires. They have this box they got to hold on to. And for the most part, they're very sedentary at home. They're asked to pretty much just sit still. If they're mobile really at all, the electrodes come off and you can't really acquire data very well. So started everybody thinking, could we really create some kind of wireless EEG for people? So it led to the very first wireless EEG that we came up with, affectionately called the dog bone. This had separation of about 10 centimeters, a really nice, interesting design with some very small electronics. Didn't really get a lot of use, just really a nice, interesting initial development. led to the next year for a much more compact sensor we called the chiclet. This was much smaller, really had very close spaced electrodes, really developed on this idea of what's known as high density EEG. The electrodes were about 17 millimeters apart, still allowed to be able to capture really good, clean EEG waveforms. The one difficulty with this sensor, as you can see in the image on the right, is that we have these holes. And the way that we got good electrical contact is you had to fill this with an electrical paste. And that was kind of messy and didn't work all that great. So, we didn't have a great way to attach this to the scalp, but it eventually led to development of the sensor we called at the time Epilogue, and is really actually the sensor that we have in use today, still 10 years later. I have some of these, and I'll pass some out and show you at the end. It's a really, really small, compact sensor. It's about the size of 3/4 stacked together. We have Bluetooth embedded in this, so we can transmit data directly off of it. But the nice design that came along with this is how we attach this to the skin. On the far right, which you can see all in white there, this is a sticker adhesive. It's really, really thin, and it allows it so that you can put the sticker on, attach it to the skin, and if you want to go take a shower, or if it's starting to degrade, you can just take it off, peel the sticker off, throw it away, put a new one on. And over the electrodes is an embedded hydrogel. And then this allows for really nice electrical contact directly over the electrodes. And this is a really nice clean design that allows nice placement of these sensors, really good adhesion, and really good low impedance contact for the electrodes. So we did all this under grant development, and still do a little bit, but this is where we really started spinning up some early human trials, all under small business initiatives, NIH and NINDS grant programs. We also at the time, this is when I started to join Epitel, we were looking at doing, really wanted this to be a long-term at-home wearable. And we wanted to bring in some algorithm development towards seizure detection, really be able to provide people some kind of seizure counting during their daily lives. And we had talked with iRhythm, the wearable heart monitoring, and they have algorithms for theirs. We wanted to find out how had they developed all of theirs. And it turns out they were able to buy data and be able to use that to help develop theirs. And we went looking for what we could do for EEG. And while there is some open source data, there's just not a lot that's well annotated, even for just when seizures are occurring, let alone where on any of the actual electrodes. And so we knew very quickly we were going to have to go develop all of our own from our own data. And this led us to really spinning up very quickly a lot of trials with our sensor from a number of clinical sites. So we needed to figure out where we could place these to be able to get good seizure activity. And one thing we had known from some early studies, one of the things I'm showing here in the far left, this is the 1020 International Electrode Placement Diagram. When a technician goes to put EEG on, this is the guidance for exactly where they have to place them. We know from some early studies that if you place electrodes just at the hat band, that outer ring, like where a sweatband would go on your head, that's a good minimum set of electrodes where you can still get good comprehensive brain activity. And we thought it could be going even less than that. Because of the way our sticker is designed, it needs to go on skin. This can't be attached over the hair. It would just kind of float up there, and you wouldn't get good actual electrical contact and brain activity. We thought, okay, can we do a more minimum set? Can we maybe get down to 4 locations? Can we get very up high in the forehead and maybe up behind the ear bilaterally, up in this FP1, F7, F3 area, and then say between T3 and T5. And when we look at what brain regions would this be able to capture, we'll still be able to basically record from frontal lobe, temporal lobe, and parietal lobe. And this is where a lot of seizure activity from the cortex occurs. As well, there's seizures that start from deep brain structures. It's believed that absence activity starts from, or is triggered from the thalamus. But these generalize. They can show everywhere across the brain, and any electrode will pick them up. We're still not able to pick up every seizure that exists. If someone has a seizure that maybe occurs very, very focal in the occipital lobe, or maybe occurs midline, we might not be able to record those. But that's not the majority of them. Midline is really just neonates, people just after birth, and occipital lobe seizures are pretty rare. The vast majority occur in these areas where we're able to record from. And we have some early data that shows this to be fairly true. We took the traditional wired data and we stripped out just the single channels where our sensors are going to be placed. And we asked clinicians, if you look at just the single channel, can you see the seizures? We knew when the seizure timings were and asked them, is the seizure visible on just that single channel? And the interesting aspect is not the individual channels alone, but more this far right column, this any one. And what we found out is if we have all four sensors, all four locations, that for every seizure that we looked at in this cohort, the 75 seizures that were part of this study, that at least one location, they were able to visualize the seizure. And this was a really interesting information to find out that, for all of these, every single seizure, that at least one location, they could see it. This told us that, this four locations, just these four alone, could be viable. So this got us thinking, we really wanted to really expand to be a medical device. But the single channel really was going to be very far development. There wasn't a pathway towards reimbursement. There wasn't a predicate system out there. But was there something we could do from the four-channel system? And one of the things that we saw is that there was a real dearth of access to EEG at what we call the point of care. If you show up to an ER and you're unconscious, it might be because you're having a seizure or something else. Or if you show up and you're having convulsions, it might be due to a seizure state or something psychological. And the only way to know is to have an EEG performed. But there's very little access to that at most ERs and ICUs. They're mostly in major metropolitan centers and really at only a few hospitals, even in those locations. So we thought, okay, could we change the paradigm a little bit from where you have these, traditional big bulky wired systems and having to have somebody right there do the reading, instead to more of a wireless sensor system and be able to transmit the data to a reader somewhere else. In this way, you could provide point of care EEG access to anybody at any of these hospitals. And, we know this exists from other arenas, from EKG. Again, iRhythm has been out for quite some time. SpO2, there's quite a bit out there. Blood glucose, Dexcom, and all these other monitors have been out for a while. And we looked at what there was for EEG, and just nothing existed at all for it. And so this led to the development of what we call our remote EEG monitoring system, or REMI. And the way this works is any ER, ICU, doc, nurse, doesn't have to be a specialized trainee, and that's normal EEG, they have to be trained. There are specialized technicians to place it. But this for our system, with very little training, can set it up in just 5 minutes. We use a tablet that helps guide them through the placement. And they get the sensors placed in just under 5 minutes. But the nice part of it is the data transmits from the sensors through the tablet up into the cloud, and then a clinician can review that data from anywhere. And the idea was we'd have some centralized place where we'd have readers, or there'd be from a hospital where then the main center, say at a major metropolitan, that the epileptologist would be able to do the read for a more rural location. And this way, again, you could have that point of care service of EEG. And it'd be better care, have treatment right away for those that are in seizure state, instead of having to transfer them to a major center. And for those that aren't having seizures, be able to triage them into the care system that they might need. If somebody's having a psychological episode, they don't need barbiturates, they don't need seizure, treatment. They need psychological care much sooner than being transported off. And this is what we developed in 2019. And this is what the data would look like for a clinician, viewing it remotely. They'd be able to see our force sensor data, as well as we create differentials, all of the longitudinal and transverse as well. So this was really the defining of our medical device that we wanted to create. And in 2020, our founder, Mark Bromkula, went to the MedTech Innovator Summit. And this is where small startups like us at the time can really meet with people that can help you find venture capital, regulatory support, and those kinds of people that can help you launch these kinds of systems. And they put us in touch with this really nice lean regulatory consultant. And we were able to get an FDA clearance that we submitted in 2020 and was cleared in 2021 with just 5 of us and this regulatory consultant working out of a 130-year-old motorcycle garage behind the bayou downtown. We were able to get this clearance for the system for in-hospital use up to 48 hours, which is fine. It's designed, again, for point of care, really rapid, know on the spot what's happening for ages 6 years and older. And that was a big turning point for us as a company. We're really small. We were down to just 5 of us running on grant funding alone at the time. And sometimes when it rains, it pours. We also hit at the time this large $7.5 million U44 translational neurotechnology grant towards developing algorithms. And I'll talk about some of those algorithms here as well. That clearance that was just created on grants alone allowed us to go fundraise. And in 2022, we closed our first Series A fundraising for $12.5 million. And that's when we rapidly grew towards becoming the company that we are now. We brought on all sorts of people, senior leadership, regulatory, manufacturing, software, all aspects. We needed help across the board. We were just 5 engineers. There's a lot that we really needed to bring on to become a real company. We've done a lot since then. We took this system that was really designed for point of care, and we've adapted it to be more of an ambulatory system, which is where we really want to go with it. More of an at-home, everyday use. We got a clearance for that in 2023, and the big aspects of that, one is the at-home utility, but a much longer use case, up to 30 days of use. And that's going to make a really big difference for being able to capture more seizure activity for people. As well, what I'm going to talk more about here as we go is the algorithms. In 2024, we cleared 2 algorithms as software as medical devices with the FDA. And as part of this, we got what's called a predetermined change control plan that allows us to update the algorithm without having to submit new clearances. And these were really new at the time. These were just starting to be part of what the FDA was allowing in 2023. We were one of the very first in the neurology space to have one, and certainly one of the only startups to do it at the time. Just this last year, we got another clearance to allow use of our system down to age 1 year and older. And we're looking at bringing it down to even younger, down to newborns here in the not too distant future. So the ambulatory system is what I want to talk about more here now. When people go to get seizure diagnoses or when they want follow-up monitoring, most of the time they have to go to what's called an epilepsy monitoring unit. And these are booked out 6 to 8 months ahead. There's very few of them out there. People have to take time off of work, take their kids out of school. They can be costly. It's a pretty big burden on people and their children. And they're small duration studies. Sometimes you can do, we talked about it, these ambulatory studies at home, but because these systems aren't that robust, the studies are very short. But people don't have seizures that often. On average, people only have a few a month. Some even have them less frequent. And it's very common that people don't have seizures during an initial study or even a secondary, and they have to go back for more. And this is where we see a really good use for our system because we can do these much longer studies in their everyday environment, there's a much more likely scenario that we can capture their seizure activity and really be able to help support their treatment. So the only real difference between the in-hospital system and our ambulatory system is really about this intermediary, this mobile device. For the ambulatory system, we use this off-the-shelf mobile phone. It's not an app that can just run on like your Android or your iPhone. We're pretty data heavy. So we use this locked off mobile platform. The nice thing with it is because it's locked off, it's always sort of running. We're assured the data makes it through. It helps people walk through sticker changes. They're using this for many days. So they're going to peel stickers off, go get in the shower, or they might want to go out for a night on the town and take it off for just a bit. They're also able to mark when they're having seizures, be able to provide us some feedback. As well, when the data goes up into the cloud, we're able to see it. And we have some customer service people that can look and see, how does the data look? If maybe they've been sweaty during the day and the data is starting to look a little off, they can reach out and help them replace the sensor. There's times where maybe they put it back on, it may not be in the best place. They can help them sort of replace it in a better location. But realistically, it works the same. Data transmits up into the cloud, clinicians can review it. And this is where we run the algorithms as well on that data. So we really launched the system late in 2024 in just a few small markets. But we also took a Series B late last year to really ramp up sales more across the country. This really is the system that we think is really the key to Epitel. And we've really started just expanding, getting it out there a lot more, just really late this last year and really pushing it this year right now. We're on track for 200 sessions this month, expecting it to go up to 500-600 by the summer, and maybe even 1,000 per month by the end of the year. We're already seeing it across all sorts of age range, and I'm going to show some interesting seizures here in a bit. We're only in a few limited clients in a few states, but we're really we're seeing growth every day right now. Okay, this is the point in the talk where I transition more towards the algorithm, the stuff that I actually work on. So we've got this amazing system that can be used for a lot of days, and it sounds great. But clinicians aren't really set up to review that much data. They're used to reviewing just a few days. And we know it can take a lot just to review that activity as it is. We know historically from talking with them, it takes about 1 hour to review every 24 hours of EEG data. And we're going to bring a lot of data in the world. You can imagine if we're doing 1,000 sessions per month, and these people might be doing 8 to 10 days per session, this is a lot of data. And this could actually be a detractor to using our system if they just have to review this and all that data. So we know, and we knew early on, that we were going to need to provide them some level of support to do this review. And we know this already occurs in other medical device space. This has been going on in radiology for a long time. There's computer vision algorithms that help radiologists know which scans have anomalies, or even within scans, where an anomaly might be. This has been done for quite some time. And we thought, okay, we're going to do the same idea. Seizures are pretty sparse in the record. Most of the time, people aren't in seizure state. It's very normal activity. So how about we create an algorithm that can highlight where in the data is most likely to contain seizure activity, really helping clinicians key in on just that data alone, really helping, one, speed up their review, but it's also possible that it might improve their actual review of it. And we know from some early data that it can actually improve their results. Back when we were trying to decide how the system might work, we had taken some early data, put it in our 10-channel montage. We took 10 patients that had seizure data, 10 patients that didn't, provided it to clinicians to review, and then we applied an early version of a support algorithm on there to the same and had them review it. And we randomized this, changed the names and everything, and they didn't know that it was the same data. And we looked at how they performed with and without the support algorithm. And that's the difference between the closed markers and the open markers. The closed markers were with the support algorithm. For the 3 reviewers, 2 of them got a little better, 1 got a little worse, but the real key here is the consensus. When we look at how they did as a whole, the star marker, they got quite a bit better. The sensitivity went up without really much degradation in false positives. They got better with the support algorithm. So this gave us this idea, this knowledge that not only can it maybe just improve their time, but maybe their accuracy would go up as well. So the real key here, though, is we're not trying to replace clinicians. This is not trying to be diagnostic. This isn't Dr. AI coming to take their jobs. The whole goal here really is support, make their world easier, allowing them time, more time to spend with patients. And I realize the irony here of that I use generative AI to do this and not an actual human when I'm talking about not taking someone's job. And I won't go into the bias that I saw, the real prompting that it took to get these images just right. So I want to talk about how the algorithm works. I'm not going to go into the really fun details. That's the secret sauce that we do. But I want to just talk a little bit about some of the key parts and how it works. In general, we follow a traditional machine learning pipeline. But one of the big things right off the bat is how we process the data to start. Because people are doing their own sticker changes and their placement, it's not always the same place every time. It's not always the same orientation. They're out moving freely in the world and the environment's changing. And you can imagine how that affects the data. So we have some really interesting math that we do to adaptively standardize the data right off the bat so that across patients and within that patient over time, we can bring all that data into sort of a very standardized reference right off the bat. And that really makes a lot of our math easier when we start there. From that, we segment the data into these short windows of time, and then we generate features of the data for those small windows. And some of this is standard stuff, like from the time domain, from the frequency domain, what's the power in specific bands, relative power, as well as things like the complexity domain. Think about entropy measures. We also do lots of additional feature engineering where we look at features of the features themselves, how the features adapt over time. Because we're doing this sort of classical machine learning aspect, feature engineering is a very key aspect for us. From those features, we do classification. What is the likelihood that this data segment is during a seizure? Now, this is where researchers tend to stop. If you look at the literature, 99% of researchers stop here. They generate ROC curves, look at something like the area under the curve, and compare themselves against everybody else out there. But this isn't useful. I can't tell a clinician that this 2-second segment's from a seizure. The next one, maybe. The next one, not. The next one, yes. That's not meaningful. What they need, and what we learned how to do, is intelligently stitch together these segmented likelihoods into discretized events. When did a seizure start and when did a seizure stop? That's what's meaningful to them. And we've created some really interesting math to do that in a nice, intelligent manner, to find these really discrete borders. I'll move this off because I want to show one other piece here. Because we have these math models to go from the segmented data to the discrete, we also know things about the likelihood that that discrete event is actually is a true seizure event. And we've created what we call this confidence metric. How likely is that discrete seizure a true event? And we add that label onto there. We start with it as a numerical number, but we've turned it more into a categorical, that's something that's a little more useful to a clinician. Low, moderate, high, and very high. And we do this to help the clinicians, but more so it's to provide some trust and a little transparency into how our math works. This way they know it's not just a blinking red light. It actually gives some context to the event that we've detected. So the way that this looks when it's presented to them, again, there's the data. but all of our detected events show up in what's called a comment window, or a list of annotations. And they can quickly click on each one of these. It'll jump to the data, and they can, look around in there, and they're the ones making the decision. Is there a seizure there or not? We're just support for them. We're just the math that's trying to help them. They can also, filter based on, maybe the confidence level if they want. But again, the data is always there. They're the ones making the decision. It's great. So we have this amazing algorithm, but we can't just put it out into use. This is software as a medical device. We have to clear this with the FDA before we can actually put it out there. So I want to talk to sort of the process of how this is done before I show what we actually did with it. So the most straightforward path is if somebody's done it before you. That makes your world a lot easier. And that's if there's an appropriate predicate, some other software that exists that's doing something similar to what you want to do. And then the idea is you show what we call substantial equivalence. You don't have to be the exact same, you just need to be pretty close, pretty similar. And the real key aspects is this idea of similar intended use, same patient population, same age, same environment of use. Similar technological characteristics. How does it work? What does it do? As well as similar clinical performance. You need to test the same methods that they do and show that you perform similarly to their results. So for us at the time, there's a very well-known software out there that's considered state-of-the-art. It was this Persist P14. But we had some really key differences. They were only cleared for use for ages 18 and above. And our system was for ages 6 and above, and we obviously wanted the algorithm to be for that. As well, they were designed for the traditional 1020 wired, which is these 16 plus channels, and we needed to be for just our 4 sensors. And we also had this really interesting novel confidence metric. So part of this is showing that everything you're doing is not any riskier. You're not creating new risks from any of these changes. So we developed the algorithm starting in 2022. We sort of finalized it in 2023. We submitted it then and got our clearance in 2024. And the way that we did this, we had a whole lot of data that we had collected from our NIH grant-funded studies, where we've been collecting our data alongside the traditional 1020 wired EEG. Some of this was from in the hospital. Some of this was from patients wearing ambulatory systems at home. These are people going under standard epilepsy monitoring, either for diagnostic purposes or after monitoring. One of the key pieces, though, is how do we get the ground truths? How are we going to compare the algorithm to something? And the method that the FDA expects is this idea of independent review and what we call consensus review. And so we took a whole bunch of independent blinded epileptologists, had them review the traditional many wired data. That's what they're used to reviewing. We gave them no other context, no video, no notes, just like our algorithm. All it's going to see is the data, and ask them to tell us exactly where do what we call electrographic seizures start and where do they stop. And then a little more to it is it focal? Is it just on, say, a single channel or two, or is it generalized across all of it? Or one other aspect we call focal evolving to generalized. Does it start local and evolve? And then the ground truths are what we call consensus, where at least 2 of the 3 have to agree that it started, and then it goes to where no longer 2 out of 3 agree. And it actually is pretty conservative, and I'm going to talk about why that matters here in a bit. But so from that ground truth set, we did that early on and created this massive pool of data that we could use for the ground truths. From that, we extracted what we call our validation set that we're going to compare the algorithm against. And we didn't just blindly or randomly pull data out of there. We really wanted to make sure that we had this very diverse representation of what we might expect the algorithm, what the system's going to see in the wild. So across age, across sex, across ethnicity, environment of use, across sites, so locations out there, across all the different seizure types. Basically, any kind of subgroup demographic that we could think of, we really worked, jengaing the crap out of this set to make sure that it was as diverse as we could make it. And ended up with this 2,600 hours of data across 50 patients, 31 of which had seizures in them. So the way we test this, the key metrics for this idea of seizure detection are twofold. The first is sensitivity. For all the known seizures, how often, how many did we get correct? And at the same time, how often are we throwing up false flags, this false detection rate? And the expectation, our necessary performance goals, we don't come up with these. These are basically based on our predicate. They set some standard and they tested to it, so we're going to test to that as well. And what I'm showing in the bottom of this is what persist P14 had tested to, and this is what we were expected to meet. So when we look at how we performed, we look across all the seizures that were in the record. Our sensitivity at the event level is 86%, but that lower bound, this sort of worst case scenario, that 95% confidence interval lower bound sensitivity for us was around 80%, much above our required 70%. Great. False detection rate, again at the overall, only around 4 false positives per day. That's kind of key. We want to make sure we're not crying wolf all the time. Really need clinicians to be able to trust us. But again, the piece that mattered, the performance goal necessity, that worst case scenario, that confidence interval, 95% corner. For us, 0.221 false positives per hour, well below that 0.35 cutoff. Again, awesome. This is our must that we had to hit, and we got there. When we look at this at a subject level, how did it work for, say, some of the individuals? Across the 31 subjects with seizures, we found all of their known seizures for 23 of them. And from the false detection rate, for 9 of them, we only had 1 false positive or less across their entire many-day record. And this was really interesting. This was our version 1 of the algorithm. This was our very first go at this. But there are areas to improve. I'm not trying to pretend this is some perfect magic math, right out of the box. There are some outliers from the false detection rate. Children and patients who are ambulatory. But this is not unexpected. You can imagine children and people who are ambulatory are moving around a lot more. There's a lot more likely artifacts in that data, and those are things that the algorithm is possibly flagging on, that we're potentially triggering on as seizures, and that's certainly areas for us to improve, as well as people with known seizures, what I call seizure positive here. It's very common that they have what we call interictal activity between actual seizures, that there's some light level activity that, while it may not have risen to the level that the reviewers flagged it as a seizure, is something that maybe our algorithm flagged on. And that's certainly areas where we can look at trying, turn down the level, I'll call it, the oversensitivity of our algorithm for those. When we look at where we missed, before we actually looked into it, we hypothesized it's likely twofold. It's likely very short duration events, and it's likely focal events where they were maybe only visual on, say, a single channel of data. And in general, that's true. We did worse on focal seizures, and it was the shorter events, but we still missed some longer duration events. In the middle panel, these focal evolving to generalized, there were some nearly 2-minute events that we missed, and these are pretty long duration events. These are ones we do want to be able to capture and should be able to. And this definitely is areas where we know we need to improve as the algorithm grows. So I want to talk a couple things, results when it comes to the confidence. The first is this idea of precision, the ratio of true positives to false positives for each level of confidence. We'd expect that if I put a very high confidence flag along with the detection, that it's very likely that it's a true seizure, and that the vast majority of all of those that we flag with that turn out to be true seizures. And so you'd expect higher precision for those. And in this fully held out validation set, it turned out to be true. The precision went up as the confidence went up. And that was really key to see that, in this set, it turned out that way. Certainly we'd like to see the low precision, low confidence, have a little better precision. We'd like to turn down that false positive rate. We still need to capture those seizures. There's still quite a bit of known seizures that we captured there, but it's still not very precise. You can imagine that in those low confidence, we're crying wolf a lot there, and we want to make sure we get better there. And the bottom there, this idea of inter-rater agreement, and when I say inter-rater here, this is how often did all three reviewers agree that there was a known seizure versus just two? And they agreed less for the same events that the algorithm had low confidence in or missed. And we'd expect this generally, that they struggled with the same events that the algorithm did. So this is one of our false positives, and I don't actually call this a false positive. Technically, yes, across the reviewers, there was no consensus. We did not have two of the three reviewers find it, but it's a clear anomaly. You can see the background activity changes, and there's some rhythmicity and some amplitude and frequency changes. Again, we're not trying to be a diagnostic. We want to make sure that we flag everything that we think a clinician needs to review. And this is something that I would think a clinician wants to review. Now, again, when we do the ground truth review, we can't ask them, hey, go mark me all the things that are maybe anomalous. That would just create a mess, and there's no real pathway to make that kind of algorithm. But we do want these to be flagged. So technically, by the actual process, this is a false positive, but I'm glad the algorithm flags these. These are ones we want it to flag. So technically, our false positive rate, in my view, is not as high as the numbers may show. And the interesting aspect was when we looked at what the reviewers showed, one of the three actually did flag the same event, but again, not two or more to actually make it a real event. And when we looked at the actual site review, they had nothing during this. So everything I've talked about so far is how we clear an algorithm through the FDA using consensus. But consensus is very conservative. And it's a fine methodology for doing the clearance. You want things that are well established as seizures. But by its nature, by being conservative, it's possible that it actually inflates how an algorithm performs. If you were to just ask an algorithm to go and compare against an individual alone, it may not perform as well. This required agreement actually inflates the sensitivity. And we've looked at this in some recent work that we've got out under review now. If I require 2 out of 3 consensus, these are less events, and they've had to agree a little more. If I go all the way up to unanimous, where all 3 have to agree, there's many less events. And what you can imagine is these are the more obvious events, right? These are the ones that they really agree on. And by being more obvious, an algorithm is likely to perform better. And the real truth is you don't have consensus out in the real world. You don't go to a clinician and they come together as 3 of them and look over your results and you get a consensus actual answer. That's not how the real world works. It's just one doctor. And so there's a different methodology of evaluating algorithms that's starting to gain some ground, and it's called an inter-rater paradigm. And the way this works is if we have, say, 6 reviewers, I take reviewer A, and I treat them as the surrogate truth. I can compare reviewer B to reviewer A. Again, reviewer A is my now temporary source of truth. And I can determine their true positive rate, their false positive rate, false negative rate against that reviewer A as truth. I can do this for reviewer C against them, and so on. And I can do this in a round robin manner, where like the next time around I treat reviewer B as the surrogate truth. And we did this analysis, and that's what's shown in these images. The bottom one is just more of a blown-in log scaled. And this is sort of a better way of comparing how are humans versus humans, as well as the algorithm directly to each human. So we had these 6 reviewers, and if you look at the bottom panel, the blue diamonds, that's the official algorithm. The low confidence is the actual detection events. If you look at how all the humans do across all their spatial variability, and you look at their farthest confidence intervals, you could sort of consider that this non-inferior to human zone. That's where the worst of humans do, is that big box border, the dashed line. And if your algorithm can work inside there, you could consider the algorithm to be non-inferior to expert humans. And while our algorithm's not quite in there in this V1, it's pretty close. The official actual algorithm is right on the cusp of that non-inferior zone. If we were a diagnostic, this would be more important, right? You'd want an algorithm that performs at the same level. You'd be non-inferior to humans. But we still think it's important for this support algorithm. We want clinicians to buy into how our algorithm works. Just because it's cleared, that means it's safe and it works to the levels that you tested. But that's not the same as proving to a clinician that it works to how they want it to work, or that it works as well as they do. And we really want to be able to show to them, that it can work as well as they do. And that's what this analysis can show to them. The two interesting aspects that we found from this, one is if we look at the moderate confidence detections, the gray markers in there, that's much more in that human non-inferiority. It's much closer to how the humans are operating. As well, there's quite a lot of variability in the human-to-human. We know they don't agree a lot. There's lots of data out there that shows this similar, but especially in sensitivity, they don't agree well on what an electrographic seizure is. And again, it's well known in literature, but this makes it really tough for us developing algorithms when they don't agree. If the truth is gray, the math is going to be gray. There's only so well that we can do. And this really kind of shows, what is the best that we can achieve from this. So this is a little more cleaned up of it. This is the aggregate. This is the average across all the co-reviewed pairs. And it just shows sort of the same idea of the data, but a little more cleaned up. And you can see, again, our algorithm isn't quite in the zone with the humans, but close. One other thing we looked at the same time is we took an off-the-shelf state-of-the-art algorithm. At this point in time was Persist 15. And again, we're operating on just 4 channels of data. The clinicians are looking at the many-wire data, and so is Persist for 15. They're operating on the 16 plus channels. And we put that algorithm on the data and compared it in the same way. And we did that to have some context. We wanted to see how well would they compare to humans. And it turns out, they very, very similar. Their point on there is really close to where we're operating. This gives us, again, a lot of confidence that even though we're this version 1, we're operating pretty good right now compared to what's the state of the art for many channel algorithm. The other real key piece from this data is this bottom right figure. The confidence metric, again, is sort of a metric of precision. And we'd expect it when we compare it against the humans. If everything was perfect, the colored line there would line up perfectly on that 45, right? The actual precision would match what we built it to show. And the truth came out pretty close. And if anything, what it shows is that we are actually a little conservative in development. And this may not necessarily be a bad thing. This, again, our version 1, as part of development, we, not purposely, but ended up training it to be slightly conservative. We're not trying to, blow smoke at the clinicians. We want them to trust these markers. And these results show that, we're pretty close to what we designed this to do. So I want to show a little real world utility. We've had this algorithm out in use for a bit now. This was a woman who had been to an epilepsy monitoring center and had no seizures during her time there. And she was sure that she was having them. And it was really sad when she had left the study that she had no data to do it. She couldn't actually start epilepsy treatment because there was no data to prove that she was actually having real epileptic seizures. So she had talked to her clinician and they prescribed her our system to go wear at home for more time. And some days in the middle of the night, this seizure was captured. And she actually didn't know the next day that it even happened. We flagged it in the record. I don't know if you can see on the bottom, but we flagged it with a very high confidence of detection. And she was able to go to her clinician. The neurologist reviewed it, confirmed that this was an actual seizure. And this was the first evidence that she finally had to prove that she was having epileptic seizures. This was a really big deal for her to finally have data to show that what she believed all along was actually happening. As well, this is some data from more recent, from a 70-year-old female. This is more moving around freely during the day. This is in the morning. And this is some what we call spike and wave activity that was happening localized on the right hemisphere. We flagged this with a moderate confidence, and again was confirmed by the clinician. We really are just starting to get some more real-world use out there. This is what we really need to know. How are we going to adapt the algorithm? And that's what I sort of want to talk sort of really near the end here is how are we able to adapt this algorithm? In the past, if you changed your math, if you wanted to retrain, we're talking about machine learning algorithms here. If you retrained it and it got better, you had to submit a new clearance. This cost money and this took time. Standard clearances take 60 to 90 days. And you don't want to necessarily do that every time you retrain an algorithm. It's really time consuming, and we're adapting algorithms all the time. We want to be able to make them better and release them in a lot more rapid manner. And the FDA has been thinking about this for many years. And a couple years ago, they released the initial draft guidance for this, again, what's called a predetermined change control plan. And they finalized the guidance this last year. And you submit this as part of your clearance, or you can submit it after as well as a separate piece. And you really lay out within it, how does your algorithm work, and what are the mechanics of the algorithm that you're going to change? Are there pre-processing steps that you're going to want to change? The classifier, are you going to plan to retrain it? And then from any of those modifications, exactly how are you going to do that? And part of it is the why. All of that really has to be well laid out. But then there's a huge process, and that's almost the bigger part of it. Are you going to get new data? How are you going to manage that data? If you're retraining an algorithm, what is the process that you're going to do that? You got to really lay all that out. How are you going to evaluate it? You obviously have this new algorithm, you have the old algorithm. How are you going to compare the two? And then how are you going to update it out into the wild? We may have sessions ongoing. You can't just, spit this out. This isn't just a website that's getting refreshed. You have to have a whole plan of how this actually gets updated into the wild. So we've learned a lot already. And the biggest thing that we've learned is the real world's very unconstrained. When we started with all of this, they're wearing our sensors alongside all the wired other things, the big boxes. And they just weren't very mobile. And we tried to do some early studies and model real world noise, but they were very small and limited. And we have people out there just wearing it for many days all across the country in the real world. And the amount of artifacts and new things that we learn is pretty much happening daily. And it's pretty amazing, the things that we're seeing and finding in the data, just that we never thought we'd see. And it's pretty fun from a data science side. Artifacts that nobody's ever really captured from EEG before that we're starting to see. These are really some of the key aspects that we know and are necessary as we adapt the algorithm. Because the system is sort of moving at a faster pace than our algorithm, we want to be able to bring it down to younger ages. Seizures can look differently, especially at very young brains. They look slightly differently in the way their morphology and the way that they show in the EEG. We want to be able to have algorithms that can support the system for these younger ages. As well, the system right now is really clinician driven. We want to do more patient focused use. The idea of alerting. Is someone actually having a seizure? Can you alert that it's occurring? Maybe very, very early on, and allow some very early treatment, potentially for a caregiver, not necessarily for the person themselves, but alert a caregiver. And then really the holy grail for this whole space from epilepsy that people have been working on for decades is what we call forecasting. And I like to call more risk management. Can you tell someone, far, far ahead what their risk profile is for having seizures and allow them to gain some agency over their lives and be able to plan more of their future? As well, there's more outside of seizure disorders. We are EEG, we're brain monitoring. There's a lot more out there. Depression, neurodegenerative diseases, Alzheimer's. There's a lot that we think we can do with our system. And as I said, our sensor's going on a decade now, and we never really thought it would get this far along. We know that it is not the end form factor, that there's a lot better ways that we can record that's a lot more user-friendly, and we've just started now developing some of that. So I want to acknowledge, first and foremost, the other four, especially Mark, our founder of modern day Epitel, that really kicked off the company that we are. The garage band put in so much. We wore so many hats in the beginning. It's about those that are in italics here that are on my data team that really helped develop the algorithm. There's a couple other algorithms that I didn't even talk about that they've really been working hard on, as well as everybody at Epitel. The modern Epitel that we are now is pretty amazing. We're now over 50 people and growing still, and it's pretty wild to see what we've become. Our clinical partners, Boston, NYU, and Colorado, getting all this data has really been magic to be able to create what we've done. The FDA, they've been great all along. They're actually very easy to talk to in developing these. But number one, the NIH. All of these translational grants have been absolutely instrumental in going from just these proof of concepts to covering all the way through clearances. They've supported us nonstop and still are. And now that we're a private company, there's people giving us much more money. We certainly couldn't hire any of these people that we have now without them. And they're not just trying to make money. These are actual healthcare ventures that really want to see these kinds of systems work across healthcare. This isn't just crypto money trying to make money. They want healthcare devices that work. And I'll end here and just say that at Epitel, we play as hard as we work. And some of our best ideas were found out in the wilderness. Thank you.
Nathan Wallace, on the predetermined change control plan, I know you probably don't want to get into too many details, but are you clear for changes to just add more data for training your algorithm, changes for your pre-processing, or what stages?
Yeah, because we were one of the very first, we actually spent a bit of time back and forth during the interactive review, and we have all aspects in there, not just more data, but fairly comprehensive of most every aspect of the algorithm we can adapt.
Thank you.
Leonardo?
Yeah, so back when you were discussing the EEG device with the component app, you described that patients are also given a specific mobile phone that's locked down and only for streaming the EEG data, right? Did you guys try to have a user device application at first and then migrate to just we're giving you a specific mobile device that's only for EEG streaming? And what were the challenges that led to that? Do you think you're ever going to migrate back to something where user devices streaming your EEG?
Yeah, good, great question. And yes, we originally tried just using a standard iPhone or Android. There's sort of two aspects. One is it's for Bluetooth sensors that are constantly streaming to it. So it's pretty heavy on that aspect. But it's also, it's nonstop data through onto there and up into the cloud. So it's pretty battery intensive. And most of us are pretty battery intensive on our devices as it is. So you can imagine if you're using something that is very battery intensive on its own, it just crushed battery life of standard devices. But this was, we constantly keep looking as devices get better. Certainly there's an option. We're even seeing, can we compress our data? Can we reduce the burden on there? There's parts more where we do more from the phone to it. All of that's a long way of saying, yes, we are constantly looking as devices get better and as we get better, can we eventually make it more of an app? The downside is when you do make an app, you need to be able to say what devices it works on. And we obviously don't want to end up in a place where it's limited. Because it's our own, anybody gets to use it, right? And we're on cellular, we're on Wi-Fi, no one's limited. The second we go to an app, it might limit use because, right, especially out in rural places, they might not have the latest device. And we might not work on, say, an iPhone 10.
Thank you.
Questions. My question is pretty simple. Why did you call it ground truths instead of verification?
Well, verification is more of creating requirements and verifying that your software does exactly what that requirement is set for. The ground truth is if my algorithm finds a seizure, I need to compare it to something, and that something is what we call the ground truth. And in general, we sort of call this the idea of clinical validation. We validate to some performance metric. The idea of verification is more some very specific requirement spec.
I saw some of your data that looked like it was discretized at about 1 second intervals. Was there a reason that was selected, and is that always the case, or is that on a case-by-case basis?
Yeah, that's a great question as well. We do it at 2-second intervals, and it is locked. That is something we're looking at making more adaptive. The more adaptive you make things, the more clinical evidence you're going to have to show the FDA that it works. The more you can lock things down, the easier it is for the FDA to understand it. This is why if you look across literature, most people say you need patient-specific algorithms. The FDA does not like patient-specific algorithms. So just because a researcher says this is how it has to be does not mean that's the best way to go, necessarily from a medical device. If you look across literature, there's many different scales of it. The idea of 2 seconds is we know brain activity, when we look at frequency space, happens in certain bands of it. And most of these tend to be in moderate to higher frequency. And I'll just say moderate as in, like 2 to say 20 hertz. Most of that can be captured in say a smaller window of time. And if you really start to expand that, you might miss dynamic changes in those frequency. If you go much smaller, you're going to miss capturing lower frequency activity. It was more of a pick. Across literature, people pick all across those small time scales.
I wanted to ask how you avoid overfitness in training your algorithm and to maintain high accuracy across objects specifically. Also, I think there's no kind that since you have so it seems that you have a lot of data. Have you considered other methods like the AI and using more agents for maybe getting a more robust accuracy?
Sure. I'll answer the overfitting first. Towards that's the idea of just a lot of data. Really making sure that in our training set, it's massively diverse. That's really the key. Obviously, as well, our validation set, we try to select it early, lock it away, shove it away. We really, we don't choose any of it based on the data. It's based on the demographics, right? We choose it, shove it off and lock it off over there. And then our training is, we have these years and years of data. We do standard methodology where we look at cross-validation throughout the training that we have. We also have some stuff that we've found from our commercial set that we look at as like an intermediate stage where we know things in there, and we take some of our early training set and look at how it performs there and can kind of gauge, is this generically working or not, long before we take it through to validation. To your second question of Why aren't I using modern models? Two answers to that. One is, I don't like modern models because they're black box. But two is, we don't need to. Unless standard, explainable, foundational math doesn't work, I'm not going to it. Okay, three answers. The FDA would much rather prefer you can explain how your math works. They're going to ask you. And so we started with that. Now we are looking at more neural network architectures. Nothing crazy. We're not trying to put transformers or any, very, very high-end math in place right away. Very tried and true. Things like LSTMs that keep history information in place, which could be key for this. That's what we're, poking at more now, but more it's prove it works better than everyday math. And until it does, we're not putting it in place. We didn't want to start there. That's sort of the short answer. It was, don't start with the black box, start with foundational math.
You may have mentioned this already, but do you have different algorithms for like different patient populations, or if you don't, are you considering that? So as you said, like a newborn versus an adult, it might have different types of seizures. Do you worry about if you create like a master algorithm, or is it really that they'll mess up one or the other, or is it really not too different where you need to split off?
There's sort of two answers to that. One is that idea of age. And that's part of why we locked it at age 6 and above. We know that, I'll just say visually, how a seizure looks electrographically. From that age and above, if I look at an absent seizure in the data, looks the same for someone who's 6 or 80. So for there, it doesn't matter. The second part is for, say, different morphologies. And I can't answer part of that. But the answer is yes. I showed a very simplistic view of how the algorithm works. Part of it I can't really dive into, and that's part of our secret sauce. But the answer is yes. We do a lot under the hood for all these different morphologies. Because yes, it's not a one size fits all. In the black box of it, it looks like that. Step back there.
We're actually looking at the seizure prevention algorithm. It could be something about it. By chance, I was going to do like that discrete start time and end time, and it not maybe point us to research paper that inspired your guys' experiment.
Sure. The sad thing is that I don't know that there was any literature out there. We knew early on that, I've pushed it since the day I started, and I didn't see anything out there. I can't tell you how ours works, but I can tell you sort of the simple frame. You can do something simple as say like a moving average, right? There's some very basic math to say, okay, I'm going to slide a moving average and I'm going to put a threshold. And when it goes above this, I'm going to start one, and when it goes below it, I'm going to stop. That was like the day one version. I'm not going to tell you what our day now version is, because again, that's a little bit of our secret sauce, but that's kind of the idea, is find some intelligent math that helps you find these discrete borders from the probabilities. Someday we'll open it up a little more. I do want to, we try to give back to the science community, but for right now, it's a little bit of our secret sauce. We're academics at heart. And at some point, we'll open up the box a little more.
You mentioned that your algorithm is too sensitive to IEDs, and would flag those like until something as big as being a seizure. And then you mentioned that you kind of want to reduce the sensitivity to those, so you're only detecting seizures, but do you not think that IEDs may be addictive, of where seizure onset zones may be, and be important for clinicians to also help them?
It's a great question, and one I think is more of a holistic question, but our system is not one for finding a seizure onset zone. We're a 60,000 foot satellite. We're not something you're going to be using to say, find a very specific seizure onset zone for a resection workup. Now, towards the point of, do IEDs matter? I don't know. But for us, and I wasn't really saying that, we're oversensitive to IEDs, more of some of these runs in there. And when I say IED, these are interictal discharges, sometimes like little spikes. We don't overflag on those, and we don't have any IED like spike detection. And it's kind of an internal struggle that, some of our sales want me to build that, and I don't believe that that's valuable for a system like ours. So some others do, but I think it's still even a holistic question in the space. Do IEDs matter? And why? Or is it like rate of spike discharge? And, for us, people out there freely moving, I don't know that you're going to really see spikes well. You might need to have a very quiet period during the night to go do it. But if somebody's freely moving and, they're out playing soccer, there's lots of artifacts that might trigger a spike detector. It's not like someone sitting in an ICU bed where it's quiet and you're going to get very clean IEDs. Our system is very different than that.
All right, yeah, we don't have any questions online, so I think we could wrap it up there. Thanks, everyone.